Hey, my name is Callie Chappelle, and thank you for tuning into this video. I'm a biology student at the University of Michigan, and this video is covering the basics for Bio 305, which is our Introduction to Genetics course. So due to the breadth of the course, I wanted to make a series of videos that highlights the most important aspects of the course. So I like to call this a quick and dirty because although this doesn't cover every detail of the course, it does give a good overview of the topics that are most important to know on an exam. So here are the topics that are covered in this video. The first is life cycles, the second is chromosome structure, the third is Mendel's laws, and the fourth is notation. So here are life cycles. You don't need to know that much about it for the class, but you should be aware that there are three main life cycles that you should know. Also, you should know the distinction between mitosis and meiosis, a topic that will be covered in the next video. So mitosis is where you go from the same to the same. So you go from 2n to 2n, 4n to 4n, etc. Meiosis is where you start with something and you end with half of that. So for example, you start with 2n and go to n, or you start with 4n and go to 2n. Alright, there are three main life cycles that you should know. The first is diploid. So that's where the adult organism and also the zygote is 2n and produces n gametes. Haploid organisms is where the adult is haploid, so n, creates a transient diploid and then produces spores that then become the adult. The spores are just n, or haploid. Finally, there's yeast. As you can tell from my poorly drawn diagram, yeast life cycle is very complicated. The big takeaway is that there are two different mating types. Alright, one topic that I have to admit sometimes I struggled with was the actual structure and terminology surrounding chromosomes. And admittedly, it's somewhat confusing. So before mitosis occurs, there is one chromatid per chromosome, okay? And here's the centromere. After replication, though, each chromatid replicates, so you have two chromatids, two sister chromatids per one chromosome. So here, before mitosis, one chromosome is just one chromatid, but after mitosis or after replication, then there are two chromatids, but still only one chromosome. All right, and then on each chromatid, if there's an allele or allele of a gene, then it's going to be the same on both sister chromatids. So if this chromosome carries the B gene, both sister chromatids will have the little b allele of the B gene. Alright, Mendel's laws. Obviously you will learn a lot more about Mendel's laws in lecture, but I wanted to cover the quick and dirty of what you need to know. So the law of segregation is where homologous chromosomes segregate equally in anaphase 1. So if we're going to draw anaphase real quick here, let's see, let's go with blue. I like blue. Actually, no, let's go with green, because I did this in green originally. So here we are at anaphase 1. Anaphase, remember the order of mitosis, prophase, metaphase, anaphase, telophase. We have sister chromatids segregating after they line up on the metaphase plate. So that just says they're going to segregate equally. So these are where we're talking about two alleles of the same gene, okay? So like let's say this sister chromatid had like the A allele, this one had the, I don't know, the big A allele. So these are segregation of two alleles of the same gene, and it's violated in non-disjunction. So non-disjunction is where this segregation simply doesn't happen correctly. So like let's say instead of this one going, these two going over to that side, they actually ended up going this way. That would be non-disjunction. Alright, the law of independent assortment, on the other hand, is that chromosomes can line 50-50 either way on the metaphase plate. So let's draw a picture of what that would look like, and let's do that in blue, because I did this in blue. So let's say that we are in metaphase 1 of meiosis, so not mitosis. So we have our cell, and we have, here are our chromosomes with sister chromatids, and this, let's see, this is big A, and this is little a, this is big B, and this is little b. So metaphase, on the metaphase plate, so here's metaphase 1. This is just saying that these can either align this way, or they can align this way, where we have our metaphase plate, and again we have our chromosomes with two sister chromatids with the same allele. We could have big A, and here where we had it lining up big B, alternatively it could be little b, little a, big B. So it has a 50% chance of lining up that way or that way, okay? And that's violated in linked genes. So what is a linked gene? That's where you have multiple genes on the same chromosome. So let's say instead of A and B being on separate chromosomes, B was actually on here. Well, obviously it can't line up 50-50 either way because A and B are on the same chromosome. Okay, so that's how the law of independent assortment is violated with linked genes. Alright, here's notation. So I've already done a little drawing on here. So the notation that you need to know is when you have a line, that means that they're on separate chromosomes. Well, when you have a line like this, a dash, I suppose it's called. Whatever's on the left side comes from the mother, and whatever's on the right side comes from the father. So here, if we have A slash big A slash little a, then we have the A allele of the A gene on one chromosome and the little a allele of the A gene on a separate chromosome. Same likewise for B. Big B, separate chromosomes. Big C, little c, big C, little c. All right. And then if you have it written this way, you can see that these are all the same chromosome because they're not separated by a dash. Oh, one thing that I didn't say up here is if it's separated by a colon, then that means that it is different genes on different chromosomes. So here we have A, B, and C. They're actually all on the same chromosome, even though they're different genes. And then little a, little b, little c is on a separate chromosome. But they're all on the same chromosome, even though they are all separate genes. Alright, the last thing that you should know is phased versus unphased. So unphased is where it's not written with this little line. Phased is where it is written with this line, and you just need to know what this means. So if I wrote this, then that is the same thing as this. This A goes with this A, this A goes with that A, this B goes with this B, that B goes with that B. Alright. So, here are heterozygotes. I thought this was pretty funny. Just allele uneven. Ha ha ha. I'm a nerd. So, hopefully you found this video informative, and you'll tune into our next video, which is about mitosis, meiosis, and non-disjunction. Thank you.